So welcome guys. Today's video is about bacteria exclusively. So you remember that in the previous video I introduced you to the world of microbes. I told you about what microbiology was about, the various branches and all of those things. But importantly, I told you that microbiology will be looking at bacteria, will be looking at viruses, will be looking at fungi, will be looking at cyanobacteria and all of that. Today, we're looking at bacteria extensively, their classification, their morphology, and the kind of structures that these bacteria possess. But more importantly, notice that not all bacteria are pathogenic, that is to say that are infectious and they can be able to cause diseases. A lot of bacteria, in fact, a heterogeneous percentage of bacteria are very beneficial to the survival of us humans and other living organisms as well, and that should be well noted. So let's look at the introduction. Prokaryotic organisms are organisms that do not have definite nucleus, and so basically these are bacteria. They have nuclear material scattered within their cytoplasm with no nuclear envelope. Their sizes will vary, but approximately they will range between 0.1 micrometers to 10 micrometers. They are ubiquitous. They can be found everywhere, in water, in the soil, in the air. They are all around and can live in diverse places. Some of them are pathogenic, like I said, others are not pathogenic, and they are harmless or beneficial in the first place. The sizes of bacteria in bacteriology, which is the study of bacteria, we would measure bacterial sizes in micrometers or the micron, and most bacteria which are of clinical importance, that means that they have infectious abilities and can cause disease, will range between the sizes of 0.2 to 1.5 micrometers in terms of diameter, and their length would approximately be from 3 to 5 micrometers exactly. Morphology of bacteria would have to do with the various shapes of bacteria that you can have. So you have bacteria that are rod-like shape. Rod means that they are longer than wide, and those are called the bacilli. Singular is bacillus. We have the cocoid shape, we have the spirular shape, which are some like bit of curly like macaroni, some bits, yeah. Now the cocci shape is a spherical or oval shape, and those constitute the major groups of bacteria. Bacilli, like I've told you, there's a rod-like shape. Vibros are shaped like comets. Spirali shape, those ones have rigid spiral forms. And note the difference between the spirular bacteria and then the spirochetes. The spirochetes, those bacteria, even though they also have spiral forms, they are quite flexible. So the difference between the spirular and then the spirochetes are that the spirochetes are flexible, whereas the spirular are rigid spirals. So that's very important. Actinomycetes are branching filamentous bacteria. We have mycoplasms that lack cell wall, and that is also very important to note. Bacteria, unlike eukaryotic organisms that have a definite nucleus and all of those things, bacteria are very primitive organisms, cannot assume the most complex forms or modes of reproduction, and so they will use binary fission in their reproduction. By binary fission, what this means is that there is nuclear division. Their nucleus splits into two, and then cytoplasm also splits into two, and so you produce two daughter cells out of a single parent bacteria, and that's how bacteria actually reproduce. So their rate of growth is by a factor of 2 in the first place, binary fission. And binary fission is actually evident in amoeba and other primitive organisms. So in terms of bacterial structure, that's a very delicate thing to talk about. You can see first the envelope of the bacteria. That's the covering, outer covering of that bacteria. An envelope will be constituted of the cytoplasmic membrane covering the cytoplasm, the cell wall that is made up of peptidoglycan. It has extracellular polysaccharides, and those are the capsules, microcapsules, and then loose slime. And note that these external extracellular polysaccharides and structures can be very, very important in determining the virulence of bacteria, whether they can be able to be pathogenic or not. And then bacteria also have appendages around, like the cilia and the pili and all of those things. Antigenic variations also exist within bacteria envelope, and that's important. So let's look at the cytoplasmic component of bacteria. So the cytoplasmic membrane, which is the cell membrane of bacteria covering them, will be made up of phospholipid bilayers with glycoproteins around. We have oligosaccharides, which are forming which are binded with the integral proteins, which allow the passage of materials in these channels and all of those things. All right. The cell walls of bacteria are made up of peptidoglycan. The thickness of the cell wall and the layers that the cell wall has will be very much important in giving the bacteria its characteristic properties, most especially whether it will be virulent or not virulent. But then another important thing is that the cell walls of bacteria can be able to determine whether they will be gram-positive or gram-negative. I'll postpone the issues of gram-positive and gram-negative bacteria to subsequent lessons, and we'll look at that extensively. But before then, let's look at a few differences between gram-positive bacteria and gram-negative bacteria. Gram-negative bacteria, in terms of their cell wall structure, they have a thin liposaccharide exterior to their cell wall. So the liposaccharides that covers their cell wall is quite thin, but that of gram-positive bacteria, the peptidoglycan layer is very, very thick. So gram-positive bacteria have peptidoglycan layer, whilst gram-negative bacteria have a thin layer of liposaccharides to the exterior of the cell wall. Effect of dye on gram-negative bacteria would be that the gram-negative bacteria do not retain the crystal violet dye and will react only with a counter stain, generally staining pink. So that's important. One thing you should note is that if you use a crystal violet dye to stain gram-negative bacteria, it would maintain the color of the dye and would react only with a counter stain, and most of the times you have a pink coloration for that. For gram-positive bacteria, they will retain the crystal violet dye and change into purple during the staining identification. In terms of antibiotic resistance, gram-negative bacteria will generally be resistant to penicillin because they contain an endotoxin called LPS, and gram-positive bacteria are susceptible to the enzyme lysozyme and also to penicillin, and so they cannot withstand lysozyme and the penicillin as well. Flagellum is, if in the instances where the flagellum is present, the flagellum has 4 supporting rings in gram-negative bacteria. These are namely the L ring, the P ring, the M ring, and the S ring. But in terms of gram-positive bacteria, they have rings that are supporting the rings of peptidoglycan layers and the plasma membrane covering as well. So those are basic differences that you can find between gram-negative and gram-positive bacteria. But like I said, we look at that extensively in a dedicated video. So bacteria structure of gram-negative bacteria and positive, if we've done that. Let's look at the extracellular structure of bacteria, extracellular polysaccharides that are found. So some of the extracellular layers, polysaccharides are complex carbohydrates. So those extracellular layers that are found on bacteria would include the capsules, the microcapsules, and loose slime. And these, where they exist, will be characteristics of the bacteria, whether they will be virulent or not. For example, bacteria generally that are encapsulated will be very virulent and can be able to cause diseases. Those that are not capsulated cannot cause diseases. The appendages that are available would include flagella, which is normally a locomotory structure, and pili, which is normally used for attachment and defense. Antigenic variation is important in determining virulence and immunity of bacteria. And so whether the specific antigen that is there can be detected by antibodies in the host organism or not will determine whether that bacteria can cause diseases or it can be immune to a lot of antibiotics. In terms of cytoplasmic components, structures within the cytoplasmic components, the cytoplasm of bacteria contains DNA. So in the interior, we have DNA, we have ribosomes, and various types of nutritional storage granules. Note that bacteria cells do not have any organelles. They are the most primitive cells that you can have. They do not have organelles. In terms of nuclear material, bacteria have nucleoid or a nuclear body, which consists of one long double-stranded circular DNA molecule, an R factor that it can use to undergo binary fission. So the intracellular material, the nucleoid and nuclear materials can be evenly distributed into daughter cells during binary fission in of that instance. So let's look at some of the cytoplasmic components again. Ribosomes function as active center for protein synthesis, and just like you've noted in cell division, ribosomes will serve as a platform doing synthesis of proteins, because then transfer RNAs will transfer free amino acids, and then mRNA will serve as the reader of the code, and then ribosomal RNAs will have to attach to ribosomes to serve as a platform for protein synthesis. Cytoplasmic inclusions such as